ThreeDRNAseq App can be applied to study RNA-seq data at levels of differential gene expression, differential alternative splicing, and differential transcripts usage. We call them 3D. The 3D app also can be used to detect isoform switches, where two transcripts isoforms reverse their relative abundances. 3D app is very easy to use. It is a web-based interface designed for biologists. The app is all-inclusive. It's not only for differential gene expression analysis, but it also improves alternative splicing analysis. It is not plant-specific. It can be applied to RNA-seq data from animals, humans, and diseases. It is very accurate and have good controls of false positives. It's very flexible to complex experimental design, such as pairwise, time series, and development series datasets. The analysis is very rapid by using the 3D app. The 3D app takes transcripts quantification data, generates expression matrix, then it preprocesses the data in various ways for 3D analysis. It does functional plots and isoform switch analysis. In the final step, users can save all the results and generate a report. The 3D app can generate publication quality figures, tables, and reports, such as up and down regression plot, Venn diagram, volcano plot, goal annotation plot, heat map, and profile plot. So, how long does it take for the whole analysis? The answer is 1 to 2 hours for beginners. I'm going to do a demo of the 3D app. The first step is to use the Summon tool in the web interface Galaxy to generate transcripts quantifications. Then the quantifications are input to the app for 3D analysis. I'm going to use the RNA-seq data from a study of Arabidopsis in response to code as an example dataset. The experiment was performed in different days at different time points and different temperatures. The RNA-seq reads were input to the Summon together with reference transcriptome ATRTD2 to generate recount and TPMs of the transcripts. Part 1. I will briefly show the transcripts quantification process. If you want to know more details, please go to the Summon and Galaxy user manuals in these links. Here is a Galaxy interface of the Europe server. The Galaxy interface has 3 panels. The first panel is a list of data tools. Once we click a tool, the second panel shows the interface of that tool. The third panel is a history panel, which records the analysis processes. Here we change the history name to 3D app. In the first step, we need to upload the RNA-seq read data to Galaxy. We click the Upload button in the top of the tool panel or in the history. In the pop-up window, we click the button Choose Local File. Then we select all the RNA-seq reads in FASTQ files from local disk and click Start button to start the uploading. We can see in the history panel, new histories are created to show the uploading process.
A few moments later.
The data uploading process takes a few minutes. Once all the history is turned in green, the uploading is done. The RNA-seq reads are paired-end. The forward and reverse reads are in separate files. In the second step, we need to create an object of data pairs. We click the button with tick symbol in the history. Tick boxes appear before the histories. We tick all the histories of RNA-seq reads. Then in the drop-down list, for all selected, we select the option of Build List of Data Pairs. In the pop-up window, we can see the RNA-seq reads are automatically paired by unique tags of forward and reverse reads. If the automated pairs are incorrect, we can click Unpair All and type in new tags of forward and reverse reads. Then we auto-pair again by new tags. At the bottom, we give a name to the paired dataset and click the button Create List. In the history, we can see a new history is created of the paired dataset. In the third step, we need to upload a transcriptome in FASTA format for transcript quantification. We click the Data Upload button again on the top of the Tools panel. In the pop-up window, we select the transcriptome FASTA file from a local disk, then click Start Upload. The history turned in green. The upload is done. Now we have prepared all the input files for the transcript quantification. In the next step, we type salmon in the search box of the Tools panel. In the search results, we click Salmon Quant. The Salmon tool interface is present in the middle panel. The interface includes different sections to select input files and set parameters for the quantification. Users can go to the Salmon user manual for details. In the analysis, in the Salmon index section, we select the transcript FASTA file we have uploaded in the previous step. In the data import section, we select the paired end datasets we have generated in the previous step. For all the other parameters, we just set to default. At the bottom of the page, we click Execute. A new history of the transcript quantification is created.
A few moments later.
Once the history turns in green, the quantification is done. We click the history to access the dataset inside the history. We can see the quantification results are generated for individual samples. Then we click the Download button on the top. The quantification collections are zipped and downloaded to a local disk. We open the zipped file. We can see the quantifications of samples are in separate tabular files. The second part of the demo is a 3D analysis. There are 3 input files required by the app. The first one is a table of the experimental design information with labels of conditions, replicates and quantification file names. The second one is a transcript quantification outputs of samples. The third one is a transcript gene association information because we need to analysis at both gene and transcript level. Here I'm going to explain the principle of 3D. In the first plot, we can see the gene and transcripts all have big expression changes between 2 conditions, C1 and C2, but the transcripts, they don't have alternative spliced percentage change. So the gene is DE only gene and the transcripts are DE only transcripts. In the second plot, the gene has no expression change, but the transcripts have significant expression and spliced percentage changes, though the gene is DAS only gene and the transcripts are DE and DTU transcripts. In the third plot, the gene has expression change, transcripts 1 and 2 have expression and spliced percentage changes, though the gene is DE and DAS gene and the transcripts 1 and 2 are DE and DTU transcripts. Transcript 3 is a special case. It only has spliced percentage change, so it is DTU only transcripts. The 3D analysis is based on the limma R package. Different statistics such as adjusted p-value, log-to-4 change, and delta PAs are used to generate significant results. Before the 3D analysis, we put the input files in the same folder. The mattable.csv is experimental design information. Mapping.csv is a transcript gene association. The salmon quant is a zip file we downloaded from the Galaxy analysis in the previous step. There are 2 methods to run the 3D app. The first one is to run the app on the James Hutton Institute server by this link. The second method is to use the ThreeDRNAseq App package in RStudio. The package can be installed from GitHub. User manuals can be found in the GitHub page. This is a 3D app interface. It has many pages. For example, the data generation, data preprocessing, 3D analysis, functional plot, other form switch analysis, and in the final step, users can save all the results. In the data generation page, in the first step, we can click the buttons to upload the import files. The first import file is the metadata table. The second import file is the transcript gene association information. The third import is the transcript quantification we generated from Galaxy. It will take a while to upload the import file, depending on the data size. In the next panel, we need to select the experimental design information. In our datasets, we are interested in the expression change of the time points at different day, so we select day and time. Then we need to select the information of biological replicates, sequencing replicates, also the quantification file names. So these tables here shows the uploaded information. For example, the map table information of the experimental design, the transcripts and gene information, the transcript quantifications. They are in separate files of the examples, so we need to put them into a data matrix. In the next step, we just click the button Run to put them into a data matrix. The next page is data preprocessing. Before 3D analysis, we need to filter the low expressed genes and transcripts. We need to remove batch effect and do data normalization. The 3D app uses different quality control plots to tell users the optimal parameters for data preprocessing. For example, to filter the low expressed transcripts and gene, we use the mean variance trend plot. If you don't know what is mean variance trend plot, you can click the question mark here to get the help information. In the mean variance trend plot, the x-axis is mean, the y-axis is variance. RNAseq reads are known to be negative binomial distribution. Their mean and variance trend should follow a monotonically decreasing trend. But in the plot, we can see the low expressed transcripts, they cause a drop end in the plot, so we need to remove this low expression part. In the app, we just set the cutoffs of the expression and then click the button to make the mean variance trend plot. After filters, the drop end is still there, then we increase the cutoff of the expression and make plot again. If the drop end is still there, we increase the cutoff. We increase the cutoff until the drop end removed from the plot. So this cutoff can be used as the optimal parameters to filter the low expression. We can have tables and we can have plot to visualize the results before and after filters. In the next step, we use a PCA plot to visualize the data variance and the batch effect. We group the examples by biological replicates and then make the PCA plot. In the plot, we can see the examples of biological replicate 1. They stay in a separate clusters compared to other two biological replicates. So this is a clear batch effect. Batch effect is caused by performing the biological replicates in different lab conditions, for example, by different people and different time. In next step, we need to remove the batch effect. After remove the batch effect, we update the PCA plot. Now in the plot, we can see the examples of the 3 biological replicates, they stay in the same clusters. Then in next step, we can do data normalization. Before data normalization, we can see the distribution of the expression are very different across samples. After data normalization, we can see the distributions are very similar. So by using this quality control plot, we have optimized the dataset for 3D analysis. In next step, we go to the 3D analysis. This table shows the condition groups of the experimental design. In the 3D app, it is very flexible to choose any condition groups we want to compare. In the next panel, we only need to select the groups we want to compare. For example, we want to compare the time group of day 1, 10.2, to the group of day 0, 10.2. If we want to do more comparison, we click Add a line. Then we select another time group to compare the another time group. In more complex cases, we can compare the mean of multiple groups to the mean of other groups, or we can compare the difference of two groups to the difference of other two groups. Then we click the button Generate Contrast Group. So in this table, we can see the contrast group information has been generated. In the next step, we do 3D analysis. In this panel, we can set cutoffs of the adjusted p-values, log4 change, and delta PS. Then we just click the button Run. After a while, the 3D analysis is done. In the next part, we use different tables and figures to visualize the significant result. For example, here is the testing statistics. Here we can type in a gene name to make profile plot. We can save the profile plot to the working directory, and then we can click the button to visualize the saved plot. If you are not happy with the dimension of the plot, you can change the width and height of the plot and save it again. Now the plot looks much better. We can have tables to tell the number of DE DAS genes and DE detailed transcripts in different contrast groups. We have the Venn diagrams. We have this up-down regression plot. We can make the volcano plot. If you are not happy with the color of the plot, you can change it and make plot again. We have other tables, other figures to show the significant result. In the functional plot page, we can make heat map of 3D genes and transcripts using different clustering parameters. And we can save the heat map to the working directory. In the next part, we can upload goal enrichment information to make goal annotation plot. In the next page, isoform switch analysis, we can set different parameters for the isoform switch analysis. Then we can use different plot to visualize the isoform switch analysis results. For example, we can type transcript names here to visualize their switch profiles. In the final page, you can click the button to save all the results. You can click the button to generate report. If the app is running on a local computer, all the results are saved in the app working directory. If the app is running on our server, you can click the button to download the results to a local disk. There are 4 folders in the saved results. The data folder includes all the intermediate datasets of the analysis. They are in R data format. Our users can access them by R command line. The figure folder are all the saved figures in PNG and PDF format. The report folder includes the final report in Word, HTML, and PDF format. If we open a report, we can see the report includes sections of detailed method, result, and supplementaries. All the important tables and figures are included in the report. The parameters and results in the report can change according to the settings in the app. The result folder includes the significant gene and transcripts list and their testing statistics. By the demo, we can see 3D app allows biologists to analyze their own datasets very quickly and easily. It's a huge save for cost and time.